Let's begin with the definition of Krebs cycle. Krebs cycle or citric acid cycle or TCA cycle is the second phase of aerobic cellular respiration. It's a cyclic reaction that occurs in the mitochondrial matrix where acetyl coenzyme A is oxidized, forming carbon dioxide, water, ATP, NADH and FADH2. Let's move into the details of the reaction. After glycolysis, what we have is pyruvate. Pyruvate enters mitochondria and forms acetyl coenzyme A. This reaction is called as link reaction as it connects glycolysis and Krebs cycle. In that reaction, NADH is formed and carbon dioxide is released. The reaction is oxidative decarboxylation and the enzyme involved is pyruvate dehydrogenase. So at the beginning of citric acid cycle, in the step 1, oxaloacetate and carbon dioxide is released. The reaction is oxidative decarboxylation and the enzyme involved is pyruvate dehydrogenase. So at the beginning of citric acid cycle, in the step 1, oxaloacetate, a C4 compound, a 4-carbon compound, combines with acetyl coenzyme A, which is a 2-carbon compound, forming citrate or citric acid, which is a 6-carbon compound. And the enzyme involved is citrate synthase. That is why the cycle is also called as citric acid cycle. So in the first step, oxaloacetate, which is a 4-carbon compound, combines with acetyl coenzyme A, which is a 2-carbon compound, forming citric acid, which is a 6-carbon compound. And the reaction is condensation reaction. In step 2, citric acid is converted to isocitric acid and the enzyme involved is aconitase. Actually, this is a 2-step reaction. First, citric acid is converted to cis-aconitic acid, which is an unstable compound by dehydration or removal of water. Then this cis-aconitic acid is converted to isocitrate or isocitric acid by hydration. The enzyme involved is aconitase in both the reactions. In step 3, isocitric acid is converted to alpha-ketoglutarate and the enzyme involved is isocitrate dehydrogenase. This is the step where the number of carbon is reduced to carbon 5. This is carbon 6. Isocitric acid is having 6 carbon, whereas alpha-ketoglutarate is having 5 carbon. Then there will be decarboxylation or release of carbon dioxide and also dehydrogenation, formation of NADH. This is also a 2-step reaction. First, isocitrate is converted to oxalosuccinic acid by dehydrogenation, which is an unstable compound. Then that oxalosuccinic acid is converted to alpha-ketoglutarate by decarboxylation or removal of carbon dioxide, forming a 5-carbon compound which is alpha-ketoglutarate. So this is a step where the number of carbon is reduced from C6 to C5. Now in step 4, alpha-ketoglutarate is converted to succinyl coenzyme A and the enzyme involved is alpha-ketoglutarate dehydrogenase. Here also the number of carbon atom is reduced from C5 to C4. So these are 2 consecutive steps where C6 is converted to C5 alpha-ketoglutarate and alpha-ketoglutarate C5 is converted to C4, that is succinyl coenzyme A. And the reaction is oxidative decarboxylation. The same thing happening in step 3, the same thing is in step 4. The carbon dioxide is released. That's why the number of carbon is reduced to 4 and NADH is formed and also coenzyme A is added. Therefore, the reaction is oxidative decarboxylation. Now we have succinyl coenzyme A. In step 5, succinyl coenzyme A is converted to succinate or succinic acid. In this step, GTP is formed. This is a substrate level phosphorylation. Succinyl coenzyme A reacts with GDP and it's a high energy compound and actually forming GTP. And coenzyme A is released, and GTP is soon converted to ATP. So this is the step where ATP is formed. Succinyl coenzyme A is converted to succinate, and the enzyme involved is succinyl coenzyme A synthetase. Here also succinate is a C4 compound. So from succinyl coenzyme A, which is a high energy compound, by substrate level phosphorylation ATP is formed and succinate is also formed during the process. In step 6, succinate is converted to fumarate. Succinic acid is converted to fumaric acid. And this is a dehydrogenation reaction where FAD is converted to FADH2, and the enzyme is succinate dehydrogenase, and this is the only enzyme in Krebs cycle that is located on the mitochondrial inner membrane. So here it is very easy to remember FADH2 is formed during the formation of fumarate, F and F. Then in step 7, fumarate is converted to malate by the addition of water or hydration, and the enzyme is fumarase, or it is also called as fumaric acid hydratase. Water is added. Now malic acid is formed. And finally in step 8, malic acid or malate is converted to oxaloacetate by dehydrogenation reaction by the formation of NADH, thus completing the cycle. And the enzyme involved is malate dehydrogenase. So this is the Krebs cycle. So it is very easy to remember if we know the number of carbon atom. Let us recap once again. A C4 compound, oxaloacetate, combines with a C2 compound, which is acetyl coenzyme A, forming citrate, which is a C6 compound. Then in the next step, then again there is a C6 compound, which is isocitrate. Then in the third step, isocitrate is converted to alpha ketoglutarate, which is a C5 compound. Then in the next step, C5 compound is converted to C4 compound whenever there is reduction of carbon number that is associated with decarboxylation and also formation of NADH. Then once the succinyl coenzyme A is formed, which is a high energy compound, that leads to the formation of ATP and also succinate in the next step, and this is a step where ATP is synthesized. Then succinate is converted to fumarate. During fumarate formation, FADH2 is formed. Then fumarate is converted to malate. It is simply a hydration reaction, and then malate is converted to oxaloacetate. These are actually regeneration steps, and malate, while forming oxaloacetate from malate, NADH is formed, therefore the reaction is dehydrogenation. So if we know the reaction, it's very easy to understand Krebs cycle. And that's about Krebs cycle.